everyone, welcome to a new episode of the Bio-Inorganic Chemistry course. Today we are going to learn about the chemistry of photosynthesis. First, let's see what is photosynthesis. We know that photosynthesis is an anabolic and endergonic process that is utilized by the green plants and few photosynthetic organisms for converting solar energy into chemical energy. The energy from sunlight drives the reaction. So, what is the reaction? You know it, you have studied in the earlier classes that the reaction is that the carbon dioxide and water molecules will combine to produce oxygen and glyceraldehyde 3-phosphate that can be subsequently converted into glucose, sucrose or other sugar molecules. What is an anabolic process? Anabolic process is that process in which simpler molecules are being combined to form complex molecules. So, here the simple molecule carbon dioxide and water is combined to form the complex molecule glucose. And it is an endergonic process that requires immense amount of energy to carry out the process. This is the equation of photosynthesis. You can see here 6 molecules of carbon dioxide and water. These simpler molecules in presence of light energy is utilized here. So, we call it as an endergonic reaction. We call it an anabolic process because simpler molecules, carbon dioxide and water will combine to form the complex molecule glucose and it will leave oxygen. So, it takes place in chloroplast of cell in presence of chlorophyll molecules. Now, let us see what is the site of photosynthesis. Chloroplast which is present in the leaves is the main location where the photosynthesis is being carried out. It is present in the mesophyll cells of the leaves. It is found around the central vacuole of the mesophyll cells. Here you can see the mesophyll cells here. It is found around the central vacuole of the mesophyll cells. In higher plants, the chloroplast is lens-shaped. It is almost 2 to 4 micrometer wide, 5 to 10 micrometer long, and you have about 20 to 40 chloroplasts per cell. Now, let's learn about the structure of chloroplast. It consists of the outer covering, which is composed of 2 membranes. It is having an intermembrane which is organized into flattened membranous sacs, which is called thylakoids. The light absorbing pigments, that is a complex chain of electron carriers and ATP synthesizing apparatus is a part of thylakoid. The thylakoids are arranged in orderly stack form which is called grana. The space inside the thylakoid sac is called lumen. The space outside the thylakoid within the chloroplast envelope is called stroma. The stroma region contains enzymes that is responsible for the carbohydrate synthesis. Here you can see the diagram of the structure of chloroplast. You can see the outer membrane, here 2-layered outer membrane. Then you have the inner membrane, you have the aqueous fluid stroma. Inside you can see the granum, which is a stack of thylakoids. This is the thylakoid. Now, let us see what are the photosynthetic pigments. Chlorophyll is the photosynthetic pigment. Chlorophylls are the primary light absorbing photosynthetic pigments. Several classes of chlorophylls are there which differ from one another in the side groups that is attached to the porphyrin ring system. This porphyrin ring is called chlorine ring system. We will learn the structure in the coming slides. Okay, we have different types of chlorophyll. First, we will see about chlorophyll A. it is present in all oxygen producing photosynthetic organisms and it is absent in sulfur bacteria. Next we have the chlorophyll B. This is the chlorophyll that is present in the higher plants and green algae. We also have chlorophyll C, which is present in brown algae, diatoms and certain protozoa. And chlorophyll D is found in red algae. Now we are going to learn the structure of chlorophyll. The chlorophyll consists of a chlorine ring, which in turn is constructed of 3 smaller pyrroline and 1 pyrrolidine ring with a magnesium 2+ ion at the center and long hydrocarbon tail. You know that it is having a chlorine ring system which is composed of 3 pyrrole rings and 1 pyrroline ring system. Most chloroplasts contain accessory pigments also. We will see what are the accessory pigments that are involved in photosynthesis in the coming slides. For example, we have carotenoids and xanthophylls, which are the accessory pigments involved in photosynthesis. So, what is the function of these pigments? The function is that they collect light energy and convey it to the central pair, that is they convey it to the reaction center of the chlorophyll by resonance energy transfer. They draw excess energy away from the excited chlorophyll molecules and dissipate it as heat.
Now let's learn the structure of chlorophyll molecule in quite detail.
We can see I already mentioned that you have the magnesium 2+ ion. This is the chlorine ring system, which is composed of 3 pyrrole ring system, that is you have having alternate double bonds and 1
Pyrroline.
That means you have 1 double bond missing here. You can see here, that is the chlorine ring system. You know that porphyrin ring system, it is present in hemoglobin, that is you have the 4 pyrrole rings. Here you have 3 pyrrole rings and 1 pyrrolidine, and the difference is that you don't have 1 double bond present here. So that is the chlorine ring system that is present in chlorophyll.
You can see the long hydrocarbon chain or the phytol tail here.
Here you can see the structure of chlorophyll A and chlorophyll B. You can see that the substituent in chlorophyll A is methyl group and chlorophyll B is the aldehyde group. Now let's see what is absorption spectra of the photosynthetic pigment. This is the graph, that is the UV spectrum of the chlorophyll pigments. Here you can see the spectrum of chlorophyll A. This one, this is the black one is that of the spectrum of chlorophyll B. You have the other accessory pigments, beta carotene, lutein, zeaxanthin, etc. Now let's see what is the importance of these pigments. These pigments are having extensive conjugation of the porphyrin ring. So what is the purpose of this extensive conjugation? It lowers the energy of the electronic transition and they are capable of absorbing in the visible region of the solar spectrum. So it will shift the absorption maximum into the region of the visible light. It also has some additional role also. It will make the ring rigid and thus less amount of energy is being wasted due to molecular vibrations. Here you can see the action spectrum of photosynthesis. So what is an action spectrum? So the action spectrum means that it is generated by measuring the amount of oxygen that is produced by leaves following the exposure to various wavelengths of light. That is,
while the light exposure to leaves is between 400 to 500 nanometer, the rate of photosynthesis is actually high.
You can see in this region 500 to 600 the production of oxygen is quite low, and here again in the 700 near region you have again higher rate of photosynthesis. So that is what the action spectrum is representing. Now we will learn about the reaction centers of photosynthesis. So here is the image of the reaction center. So what does it show? We know that a photosynthetic unit consists of chlorophyll and accessory pigments mainly. Okay. They are grouped around a central pair of chlorophyll molecule. Here we have the chlorophyll A molecule. So that is the chlorophyll A, B. So whatever chlorophyll is present, the pair of chlorophyll pigments is the central active center, and surrounding it we have other pigments. That means this reaction center is surrounded by about 200 to 400 other pigment molecules. These pigment molecules form a light harvesting antenna that absorb photons of varying wavelength, and this will be transferred by resonance energy transfer to the reaction centers by the accessory pigments. What is a reaction center? In simple words, we can say that a photosynthetic reaction center is a complex of several proteins, pigments and other cofactors that together execute the primary energy conversion reaction, that is the conversion of the solar energy to chemical energy during photosynthesis. We know that the center or the heart of the reaction center is always a pair of chlorophyll molecules. The acetyl group on each chlorophyll molecule will get coordinated to the magnesium atom to the other molecule. The sixth coordination position of magnesium atom is occupied by nitrogen from the histidine residue. So, one coordination is by the acetyl group and the other coordination is by the nitrogen atom from the histidine residue. Hope the concept of reaction center is clear for you now. So, here you can see the photosystem of cyanobacteria. There is a center chlorophyll, that is chlorophyll A here, and you have the accessory pigments. This is the 3D image of that photosystem of cyanobacteria. Now, we are moving on to the very important part of photosynthesis, that is the various phases involved in photosynthesis. Photosynthesis is categorized into two stages. The first stage is the light dependent stage and the other one is the light independent stage. The light dependent stage is again categorized into cyclic photophosphorylation and non-cyclic photophosphorylation. We will see what is it in the coming slides. So, first we will see what are light reactions and dark reactions. As the name suggests, we understand that light reactions involve the electron-proton transfer process in the presence of light energy, and this light energy is used to generate NADPH and ATP required in photosynthesis. So, remember that NADPH and ATP synthesized in photosynthesis is in the light reaction phase. What are dark reactions? Dark reactions are actually light independent reaction. That means you don't require light in this phase. In this phase, what is happening? The NADPH and the ATP that is being synthesized in the light reaction is utilized to synthesize carbohydrate from carbon dioxide and water. So, you can see it is in this phase, that is in the dark phase we are synthesizing the carbohydrate molecule. In the first phase, that is the light dependent phase, what is happening? There is synthesis of NADPH and ATP, that is the source of energy to drive this reaction. Which reaction? The synthesis of carbohydrate from carbon dioxide and water. Hope you are clear with what is light reaction and what is dark reaction in photosynthesis. Now, let us see light reactions in quite detail. Now, the location of light reaction. Where does light reactions occur? They occur in the grana of thylakoids. Hope you remember the structure or the site of photosynthesis. They occur at the grana of thylakoids. And where does the dark reaction occur? It occurs at the stroma, that is the aqueous fluid part of the chloroplast. Dark reaction occur at the stroma of chloroplast and light reaction occur in the grana of thylakoids. So, I mentioned already that in the light reaction there is formation of NADPH and ATP, and in the dark reaction this NADPH and ATP synthesized in the light reaction is being utilized for the synthesis of carbohydrate. Now, there is another process that is occurring in the light reaction. So, let us see what is it. So, here the light energy that is being harnessed in the light reaction phase is being utilized, or it is being harnessed to oxidize water. That is, water is being converted, or it is being splitted into oxygen and protons, giving 4 electrons. So, here the resulting reducing agent hydrogen or the proton will reduce carbon dioxide in the dark reaction. So, what is happening? The other reaction that is happening in light reaction is that the obtained reducing agent, that is the proton, will be utilized to reduce carbon dioxide. So, what is the reduction product of carbon dioxide? That is carbohydrate. So, it is the reducing agent is being generated in the light reaction. Here you can see in the light reaction there is synthesis of NADPH and ATP, there is the oxidation of water, and this will enter the next cycle. This is the dark phase where the carbon dioxide and water is getting converted into carbohydrate. Light reactions are also called Hill reaction. So, another name of light reaction is Hill reaction. Okay. It is a photochemical reaction that occur in the granum of thylakoids. I already mentioned that. You can see here where once more, this is the granum. It is a stack of thylakoids. The light reaction will occur here. So, it is inside these thylakoids we have the chlorophyll, and which is the green pigment that is capable of capturing light. Okay. So, the green pigment is present here in the granum of thylakoid, and it is capturing light for photosynthesis. There are also 4 types of protein assemblies which are associated to this chlorophyll, and it is also present in the thylakoid membranes and involved in the light reaction. Now, we must understand that there are 2 types of photosystems involved. So, what is a photosystem? That we will discuss in the coming slides. Okay. The first photosystem involved is photosystem 1. It includes chlorophyll and carotenoid molecules. Photosystem 2 is also having chlorophyll and carotenoid molecules. It is also having ATP synthase. It is having cytochromes B and F also. So, what is that and how does it function? That is photosystem 1 and photosystem 2, and why we are telling it is photosystem 1 and photosystem 2, where it is present. These things we will learn in the coming slides. Before that, let us see what are the different processes involved in light reactions. I have already mentioned it before, but again let us consolidate it. So, one of the process is the photolysis of water, that is the oxidation of water. What is happening there? Water is being oxidized and it is being converted into, you can see here, water is being converted into 4H plus, and we are releasing 4 electrons, and which is being utilized in the next dark reaction phase. The second process is the cyclic photophosphorylation. So, the cyclic photophosphorylation will be executed or performed by the photosystem 1. Then we have the non-cyclic photophosphorylation that is performed by both photosystem 1 and 2. So, what is photosystems and what is photophosphorylation? What is cyclic photophosphorylation? What is non-cyclic photophosphorylation? We will learn in detail shortly. The reaction center and the antenna complex will constitute the photosystem. So, that is a photosystem. The reaction center, that is the chlorophyll molecule, and the antenna complex will together constitute a photosystem. I mentioned that there are two types of photosystems, that is photosystem 1 and photosystem 2. Here you can see the processes that is involved in photosynthesis. Here you can see the reduction of water to 4H plus and O2 is generated, and these things are utilized for the synthesis of carbohydrate. Along with that, you can see here there is photosystem 1 and photosystem 2. In cyclic photophosphorylation, there is only this part, that is the photosystem 1 part, and in the non-cyclic photophosphorylation, which is present in higher plants, we have both the photosystems, that is photosystem 1 and photosystem 2 will work together. So, we will learn it in detail in the next lecture. So, once again we can look what are the processes that are involved in light reaction. The processes involved in light reaction include, the first is the light absorption. So, the light is being absorbed by the reaction centers. Then you have the water splitting, that is water is being split into oxygen and proton, and release of electrons will occur. And we have the oxygen release and formation of high energy chemical intermediates, that is ATP and NADPH. So, I have already mentioned that the byproduct of light reactions are NADPH and ATP, which is utilized in the dark phase reaction for the synthesis of carbohydrate. So, simply we can say that the processes involved in light reaction include the light absorption, water splitting and oxygen release, then the formation of the high energy intermediates NADPH and ATP. In higher plants, we have the photosystem 1 and photosystem 2 that is functioning together. In lower organisms, we have the photosystem 1 functioning alone. So, these things we will see in the coming lecture. So, in the light reaction, the solar energy is being converted into ATP and NADPH with the help of multi-pigment protein complexes known as photosystem 1 and photosystem 2. So, this is what is occurring in the light reaction. Simply we can say that the solar energy is getting converted into energetic intermediates like NADPH and ATP by the multi-pigment protein complexes which are present in photosystem 1 and photosystem 2.
Now, let us see what are photosystem 1 and 2.
In photosystem 2, it is called P680. Photosystem 1 is called P700. So, what is this P680 and P700? It means that the light absorption by this photosystem is maximum in this region, that is 680 nanometer, and for photosystem 1 the complex will absorb the maximum light energy in 700 nanometer. So, photosystem 2, that is P680, performs the light induced electron transfer reaction and photosynthesis that is responsible for water splitting into hydrogen ions and oxygen. So, the photosystem 2 is responsible for the water splitting reaction into hydrogen and proton and oxygen. The photosystem 1 utilizes light energy to generate high energy electrons which will eventually
Reduce NADP plus to produce NADPH that is required for carbohydrate synthesis in Calvin cycle.
So, the photosystem 2 is responsible for the water splitting and the photosystem 1 is mainly responsible for the reduction of NADP plus 2 NADPH, or the production of NADPH that is being required in the Calvin cycle. The PS1 reaction center, we have the reaction center as chlorophyll A, which is having the maximum light absorption at 700 nanometer. It is located in the outer surface of the grana in the thylakoid membrane. PS2, I already mentioned that it is having the absorption maxima at 680 nanometer. Where it is located? It is located in the inner surface of the grana of the thylakoid membrane. Photosystem 1 is located in the outer surface and photosystem 2 is located in the inner surface of the grana of the thylakoid membrane. Water oxidation is performed by photosystem 2. Equation we have already seen. It shows that 4 electrons are being removed from water, from 2 water molecules, and it generates oxygen molecule, 2 protons and 4 electrons. Here you can see 2 molecules of water is being split into oxygen, 4 molecules of proton is produced and 4 electrons are also generated. And this reaction is performed by the photosystem 2. We know that light reaction is also known as Hill reaction. So, what does it explain? It explains that water is the source of electrons for carbon dioxide fixation, and oxygen is being evolved as the byproduct. So, what did Robert Hill demonstrate? He demonstrated that, that's why it is called Hill reaction. Okay. Robert Hill demonstrated that isolated chloroplast evolved oxygen when illuminated in presence of suitable electron acceptor, that is like ferricyanide. So, it evolved oxygen when illuminated in presence of these electron acceptors. So, what does it happen there? The ferricyanide is getting reduced to ferrocyanide by the photolysis water. So, what he understood from the experiment? He understood that water is the source of electrons for the reduction. Okay. So, this reaction is called Hill reaction, and it clearly explains that water is the source of electrons for CO2 fixation and oxygen is evolved as the byproduct. The cyclic photophosphorylation happens only in the photosystem 1. In the cyclic phosphorylation which is occurring in the lower organisms, the only byproduct that is produced is ATP. In non-cyclic photophosphorylation that is occurring in higher plants, it involves both photosystem 1 and photosystem 2. In non-photocyclic photophosphorylation, we have 2 products, that is both NADPH and ATP are produced. So, we know that the light reaction, we have 2 phases, cyclic and non-cyclic. The cyclic photophosphorylation is performed by exclusively photosystem 1. The non-cyclic photophosphorylation is performed by photosystem 1 and 2. And the byproduct of cyclic photophosphorylation that is performed by photosystem 1 is only ATP. And the byproduct of non-cyclic photophosphorylation which is performed by photosystems 1 and 2 together is NADPH and ATP.
In this module, we have seen the introduction to chemistry of photosynthesis. We have learned about the site of photosynthesis, that is the chloroplast, the structure of chlorophyll pigment, and we have also seen the stages involved in photosynthesis. Hope you have all benefited from this lecture.
Thank you.